Hello, my name is Mitchell Slobodian, and I am a PhD student at Laurentian University, and I'll be presenting you today the metabolism of terephthalic acid in novel bacterial consortium to produce value-added products. So quick disclosure about my affiliations and funding. My work is supported by an NSERC Alliance grant, the Northern Ontario Youth Internship Program, the Ontario Graduate Scholarship, and I'm an employee of BioMine Ltd. So, we all know that plastic pollution is a major and complex issue that affects peoples, countries, and environments worldwide. And the main way it causes harm is through the weathering and breakdown of plastic pollutants into microplastics, which consequently end up inside of organisms such as fish and birds, or even us as humans in our bloodstream. To combat this, we need a sustainable, economically viable solution for plastic pollution. The main plastic I will be focusing on today is polyethylene terephthalate, otherwise known as PET, which is the most common consumer plastic in the world. Specifically, I'm going to be focusing on the monomer TPA, or terephthalate, of PET. So the objective of my work is to take our novel bacterial consortium and insert it into a culture that contains only TPA as a carbon source. We hypothesize then that TPA, that culture will grow and TPA will be consumed and reduced inside the culture and produce valuable byproducts. So to do this, we inserted our consortium into either a culture containing TPA or 10% glycerol as a control. We then collected samples and separated out the intracellular and extracellular fractions to get a look at the cell physiology and metabolomics. We then we were also able to extract RNA and DNA from both these cultures to look at the gene expression and identify the bacteria in our consortium. Right off the bat, we see an extremely interesting finding in the composition of our consortium. We can see here that our consortium is composed mostly of a bacteria called Paraburkholderia fungorum, which is a gram-negative beneficial soil microbe. Now, it's been reported in the past that this can degrade aromatic hydrocarbons, but we are the first lab to be able to show that this can degrade plastics, which is an incredible finding in the field of plastic degradation. Now, we can see here that through cell physiology that as our culture grows, we see a decrease in the amount of TPA in our culture, as well as a decrease in the pH, likely due to the production of byproducts. When we look at the metabolomics of the culture, we can see here that the main pathway that is active in this culture is the benzoate degradation pathway. The way this pathway works to degrade and metabolize TPA is through, first, by taking TPA and converting it to 3,4-dihydroxybenzoate. From there, it takes one of two different pathways to get to 3-oxoadipate, or enters into the TCA cycle to produce ATP. We then looked at these 6 genes here through qPCR to determine the gene expression. And the most interesting thing came up where we don't actually see any difference in expression within the downstream genes between our TPA culture and our glycerol control. But when we look at the very first gene in the pathway, we see a 200-fold increase in expression in TPA compared to our glycerol control. And this isn't because of a lack of CT values in these downstream genes. These downstream genes are very much active, as we can see by the raw CT values. That just suggests that Paraburkholderia fungorum, which is the main component of our consortium, has a strong enough basal expression of these downstream genes to metabolize these downstream TPA metabolites. This new bacteria has an innate capability of degrading this aromatic hydrocarbon, which is an immense finding for the field. So, in conclusion, we are able to show that our consortium is able to proliferate and survive in a liquid media containing only TPA as a carbon source, and that it grows through the benzoate degradation pathway. For future directions for this project, we plan on using CRISPR to upregulate genes in this pathway here, as well as use hi-fi gene editing to insert the genes for metase and petase to then break down and metabolize raw PET plastic. Thank you.